Hey everyone. So, this is the last video in the organic chemistry series, hopefully, and we're going to discuss biological or natural polymers, which are all condensation polymers. You remember, like condensation polymers had functional groups on them, and we used to eliminate small molecules when we made the polymers. So all the biological polymers are always condensation. There is no room for any addition polymers in these over here. So, what are biological polymers technically? Number one, they are produced by natural processes. So, they happen inside living organisms. They are produced by natural processes by living organisms like bacteria, plants, or animals. They are biodegradable. So, since they are biodegradable, which means they can be broken down by biological processes and enzymes. So bacteria could decompose them, even other algae or other microorganisms could decompose them. So there's lesser chance of environmental pollution. Starch, protein, lipids or fats, they're all biological polymers. So starting from starch, starch is made by glucose monomers. So we know what glucose is, right? Glucose is a simple sugar molecule. So starch is made by them, and it's made by a condensation polymerization where glucose monomers join together. And this is what a glucose molecule looks like. Pretty scary, right? Just focus that it has got 6 carbon atoms and it has OHs on 2 corners. And that's why we write glucose molecule as a simple box with 3 OHs on both sides. So this is first glucose monomer, this is the second one, this is the third one with OHs on both sides of all the monomers. We just remove the H and OH from each side of the molecule. So H goes, for example, from the left side and OH from the right side. It's not fixed. What is fixed is that you just remove the H and OH and you copy the whole structure. Like oxygen and the box. For the other one, you also copy the same thing from oxygen. You again copy it. Then the second thing is the box. And from the third monomer, there's one oxygen and the box. Once you're done copying it, you need to draw the extended bonds from both sides and you make the bracket and you put N. So the repeated unit is every time oxygen atom and the box. The box is actually the whole glucose molecule, but we just make it simply the box, So over here, it's not ester linkage. It is not amide linkage, not ester. It is something new that is known as the glycosidic linkage. So glycosidic linkages are present between glucose monomers when they combine by condensation reaction to make a starch molecule. This is what starch actually looks like. It's a pretty long molecule with so many glycosidic linkages. Over here, each glucose monomer is joined with the other one through a glycosidic linkage. Now, when we talk about breaking the starch molecule, that would be known as the hydrolysis. Remember, hydrolysis is breaking a longer polymer down. So over here, we can do the hydrolysis by dilute acid, any dilute acid, and then heating it strongly. So you add dilute acid on starch and then you heat it. The other possible way is a biological way, that is amylase enzyme. Amylase enzyme is the same thing that's present in our saliva. So you could use that, or synthetic saliva could be used. Proteins are made by monomers that have amine group on one side and carboxylic acid group on the other side. Remember, we used to call it amino acid because it's got amine group on one side, aka amino group, and the carboxylic acid group on the other side. Each monomer has these two functional groups on both sides of the monomer. So the H goes from the amine part and the OH goes from the carboxylic acid part. What happens is that you copy the whole structure. Starting from the nitrogen, I'll copy. There's a hydrogen on top. There's a green box. So I'll copy the box right over here. It's actually a longer molecule, but I'm just copying the box. The carbon double bonded oxygen is there. Then you copy the other monomer along with the nitrogen and hydrogen, the box, the carbon double bonded oxygen, and same thing goes for the third monomer. These monomers could be different, but I'm just copying their boxes right over here. So I've got these monomers. I can connect them and then I can make extended bonds from both sides, bracket and end. These linkages are known as the amide linkage, also known as the peptide linkage. So in language of biology, we can call it peptide linkage as well. And we have already done this video that it's amide linkage. So over here in a protein, we've got so many amide linkages between amino acids. Technically in biology, we have only 20 amino acids possible. And these 20 amino acids are arranged in so many different ways that you can have virtually so many infinite kind of proteins. So these amino acids are arranged in a very specific way to make a specific protein molecule, like hemoglobin, like myoglobin, like enzymes. So these are the amide linkages present between the amino acids. When we talk about actually breaking the peptide linkage or the amide linkage, that would be known as the hydrolysis. So if you want to do the hydrolysis of a protein molecule, what would be the way? It's same thing, right? You add dilute acid, any dilute acid, and then you heat it strongly. So you add any dilute acid and then you heat it strongly. Another way could be protease enzyme. Protease is a biological enzyme, that breaks proteins down. The third polymer is a lipid molecule. Over here you can see a very long chain carboxylic acid. We are not concerned with the long chain. So I'm just copying 3 long chain carboxylic acids down. These are known as the fatty acid molecules. And over here on the right side, I've got a molecule with 3 hydroxyl groups. So 3 fatty acids which have a carboxylic acid on each of those. So 3 fatty acids, and then on the right side of a molecule with 3 hydroxyl groups. This molecule is known as a glycerol molecule. You know what happens? The carboxylic acid loses the OH part and the hydroxyl loses the H part. And then you draw the ester linkage. So each time the carboxylic acid loses the OH and the alcohol loses the H, you get water molecule. So you get 3 ester linkages in the product and 3 water molecules as a byproduct. So 3 fatty acids join a glycerol molecule by 3 ester linkages. These are the ester linkages. When you want to break it down and do the hydrolysis of a lipid, it is same thing. You add dilute acid plus heat the lipid molecule, the fat molecule, it will break down. Or you could do one more thing. You could add a dilute alkali. You could add a dilute alkali and heat it, and then you would do the same thing. You'd break the lipid down. A biological method would be a lipase enzyme, which is a biological compound which breaks the lipids down. So this is all about the 3 most important biological polymers, and I hope the idea of condensation polymerization happening over here is clear to us. So I'll see you in another playlist, perhaps metals? Thank you guys.